Hi everyone. Today I'll be talking about my Keystone project studying muscle development in tadpoles expressing mutations for a rare neurodevelopmental disorder. I'm Lydia Bischoff and I'm a biology major, and my advisor is Dr. Isabel Dominguez, who is a researcher and professor on the BU Med campus. So the disorder I was studying over, these many years, a couple years now, is called Oker-Chung Neurodevelopmental Syndrome, or OCNDS for short. OCNDS is a rare disorder that was characterized in 2016. There are less than 200 patients worldwide, and many of these patients are children. Mutations in this OCNDS is associated in mutations with a specific gene, and many of the symptoms that the patients face are wide in range in severity, and they're nonspecific. The general theme is that they express many developmental delays and behavioral challenges. Over the summer, I had the opportunity to meet with the patients' caregivers, the medical providers, and other researchers, and I learned that many of the patients that I met with had eating difficulties. So these were challenges with being able to chew and swallow food properly. The current treatment for eating difficulties are nasogastric feeding tubes and/or changes in diet. These are huge lifestyle changes and can cause a lot of anxiety and stress for the patients and, of course, their family members and everyone involved in patient care. So our goal in the end really is to come up with a cure for OCNDS, but right now we need to understand what these mutations do to cause the symptoms that they see. I'm just looking at a small piece of the puzzle, so I'm looking specifically at eating difficulties, and our hypothesis is that perhaps these eating difficulties come about from defects in muscle development. And to do this, we will be studying muscle development in a specific model organism, and that model organism is the tadpole. And so it seems very strange to be using tadpoles and frogs. However, they share many characteristics as humans do. For example, they have the same organs as we have, so stomach, muscles, brain, heart, things like that. But they also share similar genes with us, which is very interesting, and have been used in biomedical research and developmental biology research for a really long time. Specifically, we can use female frogs lay many, many eggs, and we can have a larger sample size, and tadpoles are transparent, and so we can actually just look at them under a microscope without having to do surgery or anything like that. Also, they're easy to care for and manage, and you really just need an aquarium with a filter, so it's not too hard to take care of them. The reason why I'm spending time talking about this is because, again, it's not intuitive to use amphibians to compare ourselves with, to compare with humans who are mammals. But depending on the experiment and what we do, frogs and tadpoles are just more effective and efficient in those ways. And so I made a small little booklet for patient caregivers and just anyone who's interested in OCNDS research and why we can use frogs. Scientific literature can be a little jargony sometimes, and it can be difficult to read, and they might not explain why we're using certain model organisms and things like that. So I'm hoping this will clarify some things and make it easier to understand. And here's a little sample of what I have. So I have pictures, and I define some terms in there. So going back to my research, my hypothesis was tadpoles expressing mutations associated with OCNDS would have less muscle development than control tadpoles. And so we utilized one kind of mutation and expressed that mutated protein in the head region of the embryos. To do this, we took tadpoles at a specific age, this is right before they metamorphosize and turn into frogs, by immunostaining them to look for muscle cells in a specific muscle group. And so how immunostaining works is that this immunostain will only stain muscle cells, and the more cells you have, the more staining that should occur. And if you have more staining, you should have a brighter intensity. So they fluoresce, the cells will fluoresce under a microscope. And so you'll see brighter glowing, I guess you can call it that way. We will be using microscopes, ImageJ software, and Excel to collect data and calculate the intensities. So before I go into the results, I do want to tell you the two groups we looked at, the orbitohyoidus muscle and the quadratohyoangularis, which is quite long of a name. But these muscles are used to open and close the jaw of the tadpole. And so I figured those were extremely important if you're looking for muscle development with issues like chewing, for example. This picture on the top is actually showing the antibody staining, so it's just the green, only muscle, and the bottom just shows you it's the same tadpole, so you can get bearings of where those muscles are. So here are the results, and as you can see, the mutant-expressing tadpole definitely looks different from the control in that, like, if you're looking at the intensity of the staining, the control looks like, I consider it more like opaque, while the mutant-expressing tadpole, it looks like the muscles see through, or you can look through it. It's more darker colors, indicating that perhaps there are less muscle cells in that area. However, we can't just look at these pictures and be like, I think there's a difference. It's quite subjective. So we need to use numbers and quantify these intensities. And when we do that, generally we find a difference in mutant expressing and control tadpole intensities. However, the orbitohyoides, so that bigger muscle, had a significant difference between intensities of the control and the mutant. Why this is, I'm still not entirely sure. I do have a few ideas, but I would like to test those and learn a little bit more about that. So in conclusion, we can find that there is a difference in intensity between mutant-expressing and controlled tadpoles, and we can use immunofluorescence techniques to study this. We also need to reconduct this experiment on other types of mutations, and of course with more tadpoles. This experiment in particular was just a pilot study, and that we only had 10 tadpoles, which isn't a large sample size, and to get more clarity and significance, I think it's best to have more, definitely. However, I wanted to also say that research like this is extremely important in advancing knowledge on rare disease research. And while patients do actually grow out of their eating difficulties, because again, it's just delayed muscle development, these experiments help us better understand how do these mutations lead to these muscle development issues in OCNDS patients, and also that we could use frogs to model OCNDS-like symptoms. That's it from my presentation, but I would like to give special thanks to Dr. Isabel Dominguez, my advisor, the Boston University Kilachand Honors College, and the CSNK2A1 Foundation for all of their support and help with my project. I can take questions now if you have any.
Great job. Are there any reasons why this staining might occur that are unrelated to your research methods or unrelated to the treatment at all?
Wait, could you repeat that again?
Oh, yes. Are there any reasons why the staining might occur from the biomarkers that are unrelated to your research methods or unrelated to the processes used to treat the tadpoles?
Oh, okay. So going back to these pictures here, so the other green that you might be seeing are actually other muscles. So this specific staining stains only muscles. I just looked at a specific part, a specific group of muscles. So I wanted to look at the ones related to the jaw. However, I think in the future it'd be great to see the gut muscles as well, because you can see them in one of the pictures that I had. But I think it'd be interesting to see that too. There's a lot that we can learn.
I'm not sure exactly what your plans are post-graduation, but do you hope to continue on this research? And is this something that you see hopefully doing long-term and finding some sort of additional research for OCNDS?
I would definitely, if I can, I would love to. Especially there's so much we can learn from, of course, the tadpoles, and of course to help these patients that we're serving. So yeah.
I know this is still very early stages in your research, but do you think that as this research continues, this type of mutation is something that can be addressed in vivo or with other therapies, or maybe even genetically beforehand? Maybe it can be tested for?
I think it's possible, but I don't have a lot of knowledge on that yet. But I think learning more about this and advancing research, I think, would help a lot.
So early in the presentation, you mentioned making a pamphlet and making sure the information that you research is accessible to families with OCNDS. Do you have a method for how you might share that information? What forms might this take in sharing the pamphlet?
Yeah, my hopes is that we can submit it with the CSNK2A1 Foundation. So that is the group that brought a lot of the patients and the family members together, because having a rare disorder, it's very hard to find other people and find that support network, and it's really amazing what the CSNK281 Foundation does. I'm hoping to give it to them, and we're hoping to maybe publish it in other languages too, since there's many patients around the world with this.
Okay, if there are no questions, do you have one?
Thank you so much for your presentation. It was so interesting. I'm just curious. So you mentioned using tadpoles as kind of a ubiquitous technique in biomedical research, and now you're trying to understand like how it affects the muscles and the mutations. But I'm also curious, do you think that the tadpoles could also be used in future to test different therapies or treatments?
Yes, I think they have, and I don't think specifically for OCNDS, but for other scientific research that there has been. I don't have a specific one on mind right now, but I've heard of it, so they definitely can be.
Okay, if there are no other questions, I would like to ask one. Why didn't you use something like mice that's more commonly used in different kinds of muscular disorders?
Yeah, no, that's a good question. I think mice can be used, and they have been used to actually study OCNDS. For my experiment in particular, it seemed more efficient and effective just to use tadpoles because, again, like they're transparent, so I won't have to like do surgery on them or anything like that. So just being able to look at them under a microscope is just a simpler way just to do things. If it doesn't have to be complex isn't necessarily better. So I think for this kind of experiment, it was efficient. But mice are totally good model organisms too, and I'm not bashing them at all. So
Yeah, that definitely makes a lot of sense. Did you notice any differences between your findings and the findings in mice at all, or no?
We haven't done this in mice yet.
Oh, okay.
Yeah, no, not yet.
Okay. Any more questions from the audience? No? Well, thank you so much,
Thank you.
Lydia. That was an amazing presentation.